All right, welcome everybody. It is my great pleasure to introduce Dr. Krista Lisdahl, our first keynote speaker for the conference. Dr. Lisdahl is a professor of psychology and the director of the University of Wisconsin-Milwaukee's Brain Imaging and Neuropsychology Laboratory, the Brain Laboratory. Her primary research focus has been on characterizing the effects of chronic cannabis, alcohol, nicotine, and ecstasy use on brain structure, function, and cognition, especially in adolescents and young people. She and her lab also try to explain individual differences by examining whether genetics, gender, or lifestyle factors moderate these effects. She's been the principal investigator of over 7 large-scale neuroimaging studies and is also the site PI for the Adolescent Brain and Cognitive Development study at UWM. This is a landmark NIH study that is following over 11,000 young adults for 10 years to look at factors that affect adolescent neurocognitive development. Please join me in welcoming Dr. Lisdahl. Thank you so much for joining us.
Thank you so much for having me. I'm going to go ahead and get my slides up. Those look good? All right, gotten confirmation. It's my real pleasure to be here today. So I am Krista Lisdahl, and I'm going to be talking about adolescent cannabis use and neurocognition. And I'm going to try and get into some of the complexities of these questions as well. Before I get started, I wanted to acknowledge that here in Milwaukee, that we are in traditional Potawatomi, Ho-Chunk, and Menominee homeland along the southwest shores of Michigamme, North America's largest system of freshwater lakes, where the Milwaukee, Menominee, and Kinnickinnic rivers meet, and the people of Wisconsin, Sovereign, Anishinaabe, Ho-Chunk, Menominee, Oneida, and Mohican nations remain present. I do not have any disclosures besides NIH funding noted here. And so before I jump into my talk, I just want to acknowledge, especially with this group, that the cannabis that I'm talking about tends to be cannabis that's high in THC, and that's related to that subjective high when people are using. And over time, we've seen that THC has actually risen in kind of potency, while cannabidiol at the same time goes down. So that THC to cannabidiol ratio has really increased over the years. And there might be some opposing processes or very different outcomes if someone is using product that's very high in cannabidiol or other cannabinoids versus THC. Cannabis use really starts, especially that, using cannabis to get high in those teenage years. And this is monitoring the future data and highlighting that marijuana is the second most commonly used substance amongst teenagers. And we see here from 8th grade that those levels really rise exponentially between 8th, 10th, and 12th grade. And as noted from some earlier speakers from today, vaping of cannabis oil has also become increasingly popular. This occurs at the same time as when cortical thickness and white matter changes are happening during adolescent neurodevelopment. And specifically for my talk, we see that the prefrontal cortex and the parietal cortex are some of the last regions to really develop and mature over time. And they're becoming mature all the way into the mid to early 20s, while white matter integrity continues to improve into the 40s. THC, the main component of cannabis that I'm talking about today, is a partial agonist at the CB1 receptors in the brain as part of the endogenous endocannabinoid system. And we see over here that there is dense CB1 receptor availability in areas like the prefrontal cortex and the parietal cortex that continues to develop. And the endocannabinoid system plays a role in emotion processing, reward, executive function, memory, as well as some peripheral functions like immune and metabolic processing. And that CB1 binding actually increases from adolescence into adulthood. And it's thought to play an important role in neurodevelopment, both in that white matter development and strengthening connections and pruning weaker connections. So the theory here and kind of why I started doing this research is that repeatedly using exogenous cannabis might disrupt that endogenous endocannabinoid system, especially during adolescent neurodevelopment. And this could result in downstream neurocognitive abnormalities. And indeed, several animal studies have shown that repeated kind of heavy or chronic dosing of THC exposure in adolescent animals is associated with several downstream molecular or neurotransmitter effects. So for example, it's related to downregulation or desensitization of the CB1 receptors. It can change the structure of dendrites, such as reducing length and complexity in the hippocampus and prefrontal cortex. And it can reduce glutamate signaling and downstream synaptic plasticity that can disrupt pruning. It can stimulate microglia, such as in the cerebellum, which has been linked with reduced motor function, create downstream epigenetic changes in opioid neuropeptide genes that might be related to other drug seeking. And this abnormal glutamate and GABA signaling, as well as inflammation in areas like the frontal limbic regions, are related to functional deficits into adulthood in areas like reward, memory, working memory, set shifting, as well as behavioral outcomes like anxiety and anhedonia. In humans, we do see that regular chronic cannabis users have reduced CB1 PET binding availability in several regions, such as the prefrontal cortex here and the parietal cortex, as well as kind of paralimbic and limbic regions like the insula, anterior cingulate, and hippocampus. This does recover in about a month, although that recovery is slower if people are higher in body mass index. There is a rather large and admittedly complicated literature regarding cannabis effects. And here I'm just going to go over a few review studies just to give people a little bit of a background from folks who aren't me. The first one is looking at a literature review of notably longitudinal studies. So controlling for kind of baseline function is the onset of cannabis related to outcomes. And they concluded that cannabis leads to neuropsychological decline. The associations are generally modest, but dose dependent, and particularly in areas like verbal memory and complex attention. A couple meta-analytic studies looking at fMRI activation have found that cannabis users have abnormalities and bold response to cognitive control tasks, attention, and reward tasks, as well as functional connectivity differences and response to central executive and default mode network tasks. A recent review of structural MRI studies found that in a lot of the studies, there were 16 studies that were included, there were generally small effect sizes of those individual results. And when they were combined into a more meta regression analysis, the differences were no longer significant. These differences might have been due to heterogeneity in cannabis use patterns, sex differences, as well as MRI methods. These studies are more looking at neuropsych outcomes. So the first one in a meta-analysis of about 49 studies found small negative effect for cannabis in global and most neurocognitive domains. However, in a second kind of replication follow-up, it didn't replicate. This might have been due to heterogeneity across the studies, which was quite large, as well as big ranges in abstinence, age, and how neuropsych tests were combined. In a second, slightly more recent meta-analysis of 69 studies, they found a significant effect for learning, shifting, and I'm actually going to show this graph, speed of processing, delayed memory, inhibition, working memory, and attention, with no differences in verbal language processing, visual spatial skills, or motor skills. Notably, when they looked at length of abstinence effects, it appeared that the results were no longer significant if the users had maintained abstinence for at least 3 days. So this might suggest relatively fast recovery of function from cannabis, at least within this analysis and the studies that were included. Now I want to transition to some findings in our labs. And first I want to orient everyone to a couple of cohort studies that I'm going to be talking about. The first one included non-users, marijuana users, as well as ecstasy users, which I'm not going to talk about today. But these were young adults ages 16 to 26, recruited through ads and local high schools in the community. The youth were screened by phone, and then they came in twice into the lab with some repeated toxicology testing. After they showed at least 7 days of abstinence, they were tested with neuropsychological testing as well as structural and diffusion tensor imaging, which looks at white matter quality. The second cohort study that I'm going to talk about looked at, again, cannabis users and controls. In this case, they were actually matched on both sex at birth as well as sedentary versus physically active status. And that's related to the second aim. But the first aim was to see how cannabis affected cognition, as well as very specifically frontal limbic activation and connectivity, as well as brain structure and behavioral correlates. And the second aim wanted to look at whether or not cardiorespiratory fitness moderated these effects, which I'll talk about later in the talk. Just some more details on this cohort. They are adolescents to young adults. In this study, they came in weekly for about 3.5 weeks to confirm abstinence. And so they wore sweat patches and got repeated urine toxicology to really confirm that their THC was out of their system when they received the MRI, neuropsych battery, as well as aerobic fitness testing. So overall, there were cannabis users and non-users. A note on the cannabis users is they had to use at least 50 times a year, so about weekly. But on average, they were daily users. They couldn't participate if they were considered very heavy drinkers or had used other drugs more than 20 times, and that's also true for the controls. The controls could have used cannabis a few times in their lifetime, but not in the past year. Across both of these studies, they were excluded if they had any MRI contraindications, independent access 1 psychiatric disorders, if they are on psychiatric meds or had complicated premature birth or prenatal alcohol and drug exposure, or any neurologic or major medical illnesses. For the second cohort, where I was interested in aerobic fitness as well, they were excluded if they had any metabolic conditions, and that cohort had to maintain drug and alcohol abstinence for at least 14 days. Just some quick demographics on the sample. On average, they're about 21 years old, with just over a little bit of college. They were, as far as their verbal IQ estimates, average to kind of high average range. The cannabis users did have slightly more depressive symptoms, although they were generally in the non-clinically elevated range. They were matched for gender, although the cannabis users did have more males as far as the quantity goes. Primarily Caucasian and primarily non-Hispanic. On average, again, they use cannabis about daily. The cannabis users did have more alcohol use and were more likely to test positive for cotinine levels, although these are controlled for in all analyses. So now giving an overview of our cognitive, structural, and fMRI findings in these cohorts. This first one is to just kind of orient everyone to a couple of cognitive studies that were done kind of early on, trying to understand the cannabis effects. This first one is actually with Susan Tapert's data, finding that adolescent cannabis users compared to controls after a month of abstinence did have significant deficits in psychomotor speed, complex attention, story learning, and and sequencing ability. And these were kind of small to medium effect sizes. When we replicated this in our young adult cohorts, we found dose-dependent relationships with past year cannabis use. They had been abstinent for 7 days, and we found reduced psychomotor speed and sequencing ability, sustained complex attention, and cognitive inhibitory control. And these effect sizes were slightly smaller in the young adults compared to the adolescents. This next study is done in collaboration with Dr. Janessa Price, who's pictured there. And this was looking at brain volume, especially in prefrontal and parietal regions. And the cannabis users had reduced volumes in the medial orbital frontal cortex, as well as the inferior parietal cortex. And these medial orbital frontal reduced volume was related in a dose-dependent way with their cannabis use. These findings were generally small to the medium effect size. In a follow-up study with the former graduate student Schuyler Schoonbelliger, we saw that cannabis users compared to non-users had reduced what's called local gyrification index, or kind of the complexity of the cortical folding in prefrontal regions, including the medial prefrontal, frontal poles, and ventral medial prefrontal cortex. And this reduced kind of gyrification complexity was also reduced or linked with reduced working memory in this cohort. In the next study, we looked at white matter quality in similar frontal limbic regions, and we found that the cannabis users had reduced white matter integrity in this study measured by increased mean diffusivity in frontal limbic tracts such as the uncinate fasciculus bilaterally, as well as the forceps minor. And in this cohort, reduced white matter integrity was related to increased depressive symptoms and increased self-reported symptoms of apathy. So this leads into a series of studies that really looked at either fMRI bold response to a task or connectivity at rest. In this case, it's a resting state connectivity study focused on the default mode network. And this was done with Megan Ritchie here pictured. And she found that the cannabis users had reduced bold connectivity between the posterior cingulate and default mode network hubs, such as the precuneus and the perihippocampal gyrus. This reduced connectivity between default mode network hubs was related to poorer selective and sustained attention processing. She also found increased bold connectivity between the PCC and cerebellar regions, and this increased connectivity was related to reduced sustained attention and poorer verbal learning in this cohort. This pattern suggested that regular cannabis use might disrupt default mode network connectivity patterns, both within and between default mode network and hubs and the cerebellum, even after 3 weeks of monitored abstinence. This led us to really want to look more at other attention networks. So Julia Harris, who's pictured here, looked at whether or not there were functional connectivity differences, again, at rest between the DAN, the dorsal attention, and the VAN, the ventral attention networks. What she found was that cannabis users had reduced connectivity in the DAN network between the right inferior parietal sulcus and the anterior insula. This was linked with a few different markers of cannabis use severity. So decreased connectivity between these regions was linked with increased frequency and quantity of cannabis use in the past year, shorter lengths of abstinence, and increased cannabis use disorder symptoms, which you can see here in this plot. We did not find differences in the ventral route. So the DAN is a top-down frontal parietal network for visual attention control and processing, and disruption might lead to complex attention problems in cannabis users, which we've measured in different studies as well as this cohort. Our next study wanted to look at inhibitory control, so that ability to kind of stop a response or an automated response. We utilized an emotional no-go task, and this particular study by Alex Wallace, who's pictured here, looked specifically at stopping your response or not going during neutral or calm phases. He did not note any differences in performance across the groups in their behavioral outcomes, but the cannabis users had increased bold activation, and this was a whole brain analysis study, and left medial superior frontal gyrus and left cingulate cortex. So this pattern suggested that after those 2 weeks of abstinence, there was greater prefrontal resources needed to complete the same inhibitory control task. And this might be showing some successful compensation during that period of abstinence. In another study of kind of intrinsic resting connectivity patterns, Dr. Schoenberger and I looked at cannabis users in the IDEA consortium. And we found that cannabis users had increased bold connectivity between the left rostral anterior cingulate and the right rostral anterior cingulate, left amygdala, left insula compared to non-users. These are cognitive control and affective processing regions, again, at rest when they're not engaging in a task. And this increased intrinsic connectivity was linked with greater depressive symptoms in this cohort. And cannabis use might disrupt limit connectivity patterns, and this is associated with downstream mood symptoms in this cohort. The next study is actually getting a little bit more into that emotional processing during tasks, and also some studies looking at that interaction with no-go. These first two studies were really focused on the rostral anterior cingulate and how that might link with fearful face processing or emotional processing in general. And this was done by Kristen Maple, pictured here, as well as Ryan Sullivan. The first study found reduced left rostral anterior cingulate volumes in cannabis users, pictured here, and that was linked with worse emotional discrimination. So smaller volumes related to poor emotional discrimination scores. She also found in a region of interest that there was reduced rostral anterior cingulate bold response during fearful face processing during kind of the go condition of the no-go task, but there were no connectivity differences. So after 3 weeks of abstinence, the cannabis users had reduced volume in the rostral anterior cingulate as well as blunted bold response. And this was linked with worse emotional processing. And it might suggest that there could be an interaction between affective processing and emotional control. The next study done by Ryan Sullivan that's currently in revision focused in on both ventral striatal regional bold response and whole brain analysis examining responses to happy faces while people were engaging in go and no-go portions of the fMRI task. The cannabis users demonstrated after 3 weeks of abstinence, while engaging in happy go responses, increased bold in the left cingulum, as well as decreased bold response in the pre-SMA, which is more of a motor processing area. In the region of interest analysis, he found actually increased bold activation in the ventral striatum to those happy go faces. So cannabis users had increased reward and emotion processing response, and this was consistent with prior reward studies. In that same analysis, he looked at responding to those happy faces when they had to inhibit or no-go their response. And in this case, the cannabis users actually showed diminished bold response in the left superior frontal region, pictured here. Notably, healthy teens typically during this task actually recruit more prefrontal areas when processing these appetitive faces. And in the ROI analysis, he found blunted ventral striatal response in the cannabis users, pictured here. So they were kind of blunted when engaging in inhibitory control, even though they're processing these appetitive faces. Whereas when they didn't have to engage in inhibitory control, they actually had exaggerated bold response in that same region. So there might be an interaction between inhibitory control and affective state related to cannabis exposure. Although notably, we definitely question whether this is a consequence of or a risk factor for escalating cannabis use during adolescence and young adulthood. We also have looked at some moderators of cannabis effects, and I just wanted to highlight a couple studies that focused on whether or not you have a diagnosis of ADHD as a potential moderator. For those who aren't that familiar, ADHD is attention deficit hyperactivity disorder, and it's highly comorbid with cannabis use. In fact, cannabis is really the drug of choice in teenagers and young adults who have ADHD. So in collaboration with the MTA study, which is a childhood cohort study that actually looked at in childhood the effects of different behavioral and medication interventions, those kids came back when they were in their young adult years here, so on average about age 23, and got an additional deeper phenotyping of their substance use, as well as some brain MRI and neuropsych testing. So the goal was to look at the impact of cannabis use and the interaction with persistence of ADHD symptoms into young adulthood using that multi-site MTA longitudinal study. For this study, we looked at hippocampal, amygdala, nucleus accumbens, and cortical thickness outcomes. A little bit about the sample. It included folks who did not have childhood ADHD, that did and did not use cannabis, as well as those with ADHD and comorbid cannabis use. One thing that's notable is that the youth who had ADHD and were using cannabis did have an earlier age of cannabis use onset by almost 2 years compared to the non-ADHD controls. And they are primarily male sample. So in that structural MRI study, we found that regular cannabis use, so at least weekly, was linked with reduced left hippocampal volumes and reduced cortical thickness, especially in frontal and parietal regions, pictured here. So the superior frontal, as well as anterior and posterior cingulate. ADHD was also related to cortical thickness outcomes with reduced precentral and postcentral thickness. But cannabis use was uniquely linked with frontal and parietal structure above and beyond ADHD diagnosis. Also, earlier age of cannabis use onset was uniquely linked with increased nucleus accumbens volumes, as well as right superior frontal and postcentral gyrus cortical thickness. Notably, the main effects of cannabis on cognition in a similar overlapping sample weren't significant after controlling for ADHD diagnosis, but early age of cannabis use onset was linked with worse executive functioning. So this early onset might be related to increased sensitivity to drug rewards with some changes to the nucleus accumbens, and it might also be related to with a more kind of immature cortical thickness pattern with delayed pruning with these greater cortical thickness in superior areas. Alex Wallace, pictured here, is also interested in kind of the comorbidity between ADHD and substance use. And he noted that even subclinical ADHD symptoms might potentially explain previous findings in cannabis users. In the cohorts that I've described for my studies, we actually excluded independent ADHD diagnosis, but we did get some symptom counts of ADHD symptoms. So he wanted to know, after these 2 weeks of abstinence, does cannabis use and ADHD symptoms independently or interactively relate to cognition, especially in complex attention? And he found that cannabis use after controlling for these ADHD symptoms was related to slowed continuous performance task hit rate time, but also some decreased commission errors. But the ADHD symptoms and the interaction with cannabis were not significant. Now, notably, future work is really needed to tease apart this interaction of cannabis use and ADHD, both diagnosis and symptoms across development. My lab has also been interested in whether or not aerobic fitness might moderate the effects of cannabis. So why aerobic fitness? It might sound a little random, but I was looking for something that is, behaviorally common and might be linked with brain health. And aerobic fitness has been linked with verbal memory, executive functioning, and brain structure, especially in older adults, although less is known in adolescents or young adults. Aerobic exercise, very interestingly, releases endocannabinoids, which cannabis, of course, interacts with and is thought to be the underlying mechanism through which there might be changes to the brain during adolescence. It also has several other positive brain effects through the release of vascular and growth, brain growth factors. So our aims here were to examine whether aerobic fitness was directly linked with neurocognition and whether it moderated effects of cannabis on neurocognitive outcomes. In one of the first studies published here with former graduate student Natasha Wade, pictured here, she found that increased cannabis use was related to reduced working memory, sequencing, and psychomotor speed after 3 weeks of abstinence in this cohort. And also, interestingly, aerobic fitness was linked with better visual memory, verbal fluency, and sequencing ability. And notably, this is in young, healthy people without metabolic conditions which typically the older adult studies do include things like diabetes or other metabolic conditions, but they're very healthy in this cohort. And aerobic fitness moderated cannabis effects on executive functioning and psychomotor speed, in that highly fit cannabis users had better cognitive functioning compared to low fit users. In a follow-up study done here by Ryan Sullivan, we looked at a couple different moderators of structural MRI outcomes. So in this case, we're looking at whole brain volume. And notably, the main effect of cannabis is not significant. But when we looked at cannabis by sex effects, we found significant interactions predicting left lateral orbital frontal areas, inferior temporal, precuneus, caudate, and superior frontal, as well as right paracentral volumes. The cannabis using males compared to non-using males had smaller regions, and the cannabis using females compared to non-using females had larger regions. The effects were generally more robust in the males, and follow-up brain behavior relationships found that these were generally not advantage for either group. So, for example, the cannabis using females having larger volumes was not linked with better cognitive function. We did find that aerobic fitness, increased aerobic fitness was related to increased volumes in frontal, parietal, cerebellar, and caudate regions. And there was one region that interacted, that there was interactive effect between cannabis and aerobic fitness, and that was the left temporal volume. And in this case, the cannabis users had a less robust relationship versus controls. But overall, both the cannabis and non-cannabis users demonstrated this positive effect of fitness on brain health. And it might be that exercise could lessen negative consequences on brain health in regular users. Another big question that we've wrestled with, both in my lab as well as other labs and in the literature, is whether or not there's recovery of function with sustained abstinence from cannabis. This is a little bit of a busy slide, but this is actually collapsing, I guess, almost 20 years of research. And over here, I just want to orient you to studies that really looked at cannabis users compared to non-users that required only about 1 to 3 days of abstinence. So we could say in these studies that the cannabis users are no longer high, but they probably have THC in the system. And indeed, there could still be some kind of THC even at the receptor level. And in those studies, we found several different cognitive deficits, like even an IQ or things like verbal IQ, which we typically don't find with more abstinence, verbal memory, executive functioning, working memory, complex attention. We see differences in structural MRI, especially between prefrontal, parietal, limbic, and cerebellar regions. And we do see some abnormalities in fMRI, especially in attention, spatial working memory, executive functioning, reward, and default mode network. If we move out to requiring at least a week of abstinence, and then during this period, we do see that most of the acute withdrawal is significantly diminished, although not completely resolved. And, for some of the users, THC might be out of their system, but we wouldn't expect that there's full recovery of withdrawal symptoms or that THC is out of everyone's system. And what we see is that there still are some verbal memory, working memory, complex attention, and processing speed differences, although notably about this time, we see significant recovery in longitudinal designs in verbal memory. Structurally, we continue to see those prefrontal, parietal, and limbic differences. If we go out to studies that really required at least 3 weeks of abstinence, we find that there is some recovery. It's a little mixed, like some areas of complex attention significantly recover during that period, while others stay significantly worse in cannabis users. There is starting to see some processing speed improvements in longitudinal design. Working memory in some studies is still impaired. The structural findings, might be very subtle and be moderated by sex and aerobic fitness and things like that. But the main cannabis effects remain in areas like the rostral anterior cingulate and the hippocampus. And we do most notably continue to see fMRI differences, both in task activation as well as connectivity patterns. Notably, during this time, withdrawal is for the most part complete, and the CB1 receptor kind of availability might have recovered. There's something missing on this graph, is there's very little information, that really goes out 4 weeks and beyond. And so hopefully this recovery would continue with ongoing abstinence, as we see in other models, such as recovery of dopamine availability with 2 to 3 months of abstinence in cocaine, methamphetamine users, and as well as ongoing recovery of brain structure in people who use alcohol heavily. We just don't have, we don't have that much data in cannabis users, and people that are out there might correct me if I'm missing it, but I have not seen any repeated longitudinal abstinence studies that incorporated structural functional MRI or MRS to really look at recovery of fMRI processing. Also, this recovery in cognition that we're seeing noted here in kind of downstream neuropsychological testing, there's a question as to whether or not that's true full recovery or maybe showing successful compensation. Because, for example, in my cohort, we see a significant improvement in complex attention over 3 weeks. However, we're continuing to see, subtle abnormalities on fMRI testing, including in attention-related networks such as the dorsal attention network and the default mode network. So overall, what I have found across multiple studies now is that repeated recent cannabis exposure during adolescence and young adulthood has been linked with modest abnormalities in cognitive function, especially areas such as verbal learning, complex attention, psychomotor speed, and to a lesser extent, inhibitory control, as well as subtle changes or differences in structural MRI, especially in prefrontal, limbic, and parietal regions, followed by abnormal bold response in attention networks, default mode network, fear emotion processing, reward processing, and inhibitory control networks. And these findings, at least in my studies, are after 2 to 3 weeks of monitored abstinence. Across multiple studies now, myself and other people have found the greater frequency of use is most detrimental. And that adolescents and as well as young adults who have an earlier age of onset seem to be more vulnerable to these neurocognitive outcomes, especially compared to older young adults and certainly full adults. Sex might moderate some of these effects, though not all. Several of these studies looked for sex moderation and didn't find it. But when we do find it, we tend to find that male users might have greater effects. And a lot of studies haven't really dug into why that might be. One of my hypotheses is that males tend to be heavier drinkers and that there could be some effects of co-use here that I didn't really get to talk about today. Across most of these studies, the effect sizes are generally small to medium, so between 5 and 15% differences. And meta-analyses that really combine studies across methodologies generally report small effect sizes, and some are even null, although all of them report great heterogeneity across studies, which is difficult to incorporate when you're doing meta-analyses. I think it's really important to note that there's hopeful evidence that some of these neurocognitive deficits might recover with abstinence, and some areas might recover quite rapidly, even within 3 to 14 days of sustained abstinence. Although notably, we need longitudinal MRI research to really confirm some of that. And finally, aerobic fitness might serve as a potential cognitive rehab or even prevention option to kind of minimize some of these neurocognitive effects. I think it's really important also to talk about the clinical implications. On one hand, we have some evidence here where cannabis users look different than controls, But as a clinical neuropsychologist, it's important to note that the cannabis users are often still in that average or even low average range. Usually at worst, they're more like in the mildly impaired range, and this is being tested after abstinence. And there might be some rapid recovery of function, or at least some very effective compensation with even 1 to 7 days of abstinence. But I still have to ask, are they disrupting their full potential? So many do not want to quit. In fact, the research is pretty clear on that. So they might be high during schoolwork or at school or at work, depending on their age, and acute effects are typically worse. There are studies showing that loved ones report everyday problems in folks that use cannabis regularly, especially in attention and memory. And other longitudinal studies have noted areas such as reduced resilience, increased negative emotionality, and decreased social reciprocity in teenagers who start at a young age in longitudinal designs. And epidemiological studies have found reduced GPA, educational achievement, income and employment, and life and relationship satisfaction. So kind of taking all of those things together, it's really recommended that, especially adolescents might delay their cannabis use or reduce how much they're using. Another big issue in the literature that I'd like to acknowledge is that it's hard, or we cannot determine causality with cross-sectional studies. And there are longitudinal studies, but a few of them that did prospective longitudinal design that were twin-informed found that even though cannabis use did predict poor outcomes, such as poor verbal IQ score in this case, when they controlled for twin status, that result was no longer significant. Now, there were some methodological concerns about these studies, such as there wasn't very fine-grained cannabis use measurement, and they didn't look at other measures that might decline, such as memory, processing speed, and they were really quite small samples once you incorporated the twin part of it. But it really highlights the need for large prospective longitudinal studies. And that leads me to the Adolescent Brain Cognitive Development study. And when I was introduced, it was said that I was the site PI along with Chris Larson at UWM for the ABCD study. And this is a nationwide multi-site study that's following 11,875 youth who are at baseline ages 9 and 10. We're following them for at least 10 years. So this is a national diverse sample that's twin informed. And we're looking at multiple factors that influence brain, cognitive, emotional, and health development. And notably, this is an open science model, so data is released on a yearly basis to the scientific community. And this cohort was assessed prior to the onset of cannabis and substance use, beyond some caffeine use and a little bit of alcohol sipping. This is just highlighting the kind of ABCD timeline. So they start at ages 9 and 10. We're following them yearly, and every other year they have very deep phenotyping of cognitive function as well as neuroimaging and bioassays. The substance use assessment, I'm one of the substance use assessment co-chairs. We're looking at detailed characterization of substance initiation and use patterns, as well as substance use attitudes like expectancies, perceived harm, how available it is. We look at substance use in their environment by their peers, siblings, parents, as well as secondhand exposure. And if they're using substances, we look at consequences of that, such as substance use diagnostic symptoms, diagnosis, as well as withdrawal symptoms. And we are collecting biological samples to look at toxicology. There are a lot of questions. I'll just go over a few of them that I'm interested in. First off is replicating findings in a larger, more diverse sample, determining to what extent neurocognitive abnormalities are premorbid as a risk factor for cannabis use or really due to direct cannabis exposure in a prospective longitudinal design. We also want to start teasing apart the impact of product type, potency, content, and drug co-use. I'd like to examine the impact of cannabis use on circulating endocannabinoid levels in a smaller subsample to look at that mechanistic change. And importantly, we need to identify at-risk and resilient factors for both the development of cannabis use disorder as well as neurocognitive outcomes. And within that framework, really consider biological, environmental, drug policy, family, peer, and cultural factors that play into those trajectories. and really look at the interplay between cannabis and psychiatric disorders in a lot more detail. And finally, we'd like to determine whether sustained abstinence results in returning to that neurocognitive trajectory they were on before any cannabis use was initiated. And before I wrap up, I just want to acknowledge that this is team science, and I have a really phenomenal group at UWM in the Brain Lab, pictured here, as that includes faculty, graduate students, and a lot of extremely talented undergraduate students. So if you get applications to graduate programs and med schools and they come from our program, know that they worked on a really great dynamic team and learned a lot from the ABCD study. And of course, I want to acknowledge our funding from especially NIDA and NAAA. And I will leave it there. I had a hard time tracking questions. I saw that there were red boxes popping up, so.
Yeah, so if you do have questions, please try and put them into the Q&A function. I will also take a look at the chat and see if we can find any there. The first question comes from Margot. Would it be true that for ADHD patients, high dose chronic cannabis would actually make baseline ADHD symptoms worse?
Generally speaking, the data so far would suggest that. So both ADHD and cannabis use were associated with poorer complex attention and executive functionings. And this was particularly true in the case of ADHD if people had used before the age of 16. And we did see that that was more common in folks with ADHD. Now it's a real double-edged sword, because when people use cannabis, it really calms down hyperactivity. So, you know, I've definitely had a lot of people point out that they feel much better as far as their kind of irritability and hyperactivity symptoms, and that's why they prefer cannabis. But it does not look like it's doing them any favors for complex attention and executive functioning in particular.
Great. Next question from Sue. What was the average THC % content in users in these studies?
That's a great question, Sue. I wish I knew. So we did have sweat patch technology that gave some quantified THC levels that we just were more tracking to make sure that was out of their system, but we didn't collect product and really test it. We just didn't have those capabilities. So all I can really do is map it on to like average DEA seizures and how people kind of report the potency. So most of them thought that they were smoking, you know, medium to fairly potent product and using it to get high.
Great. Another question. Have you looked at the effect of cigarette smoking? I read one study in adolescents where controlling for tobacco smoking erased the difference.
Yeah, that's a great question. There are some studies when you control for nicotine use, the results go away. And these cohorts, cotinine or nicotine use was controlled for in all analyses. So all reported results were above and beyond any nicotine or alcohol use. But we have seen that nicotine itself is linked to some subtle attention, like especially complex attention results. In this case, like the most of the fMRI findings that I talked about today, the nicotine use is actually very, very low. So the earlier study that looked at structure and DTI did have more nicotine use in there, and we still did find differences above and beyond the nicotine use.
Next question from Tony George. Krista, chicken or egg? Are the modifiable cognitive deficits in ADHD pre-existing or cannabis related, or both? Are there studies of extended cannabis abstinence in ADHD persons?
You answered, Tony. I see Alex Wallace is in the attendees, and he has really looked at this in more detail, even in like the ABCD cohort of trying to look at, you know, what predicts what. And even finds like very early initiation is linked with worsening of ADHD symptoms, for example. And so my best guess right now is that they're complex interacting factors. Usually childhood ADHD comes first, like the diagnosis developmentally. And then there's a preference for cannabis, I suspect, because it helps kind of calm the mind and feel less hectic and hyperactive when people are using acutely. But the problem is that the endogenous endocannabinoid system plays a role in that complex attention. So if you use too much of it and you downregulate that system, you start to take a hit in the attentional and executive functioning networks, ultimately making symptoms worse. So that's my hypothesis, that it's interacting over time. But we really need to look at this in a lot more detail, because frankly, the ADHD longitudinal cohorts typically didn't look at substance use very deeply, and the substance use studies haven't looked at ADHD very deeply. So we really need to look at it at the same time, like in the ABCD cohort.
A couple of questions from Tom. First question: from your data regarding overall impact of regular cannabis use on the neurocognitive function of teens, doesn't even the term moderate deficits imply disruption of significantly large numbers of neurons? And I wonder if this needs to be stressed when putting knowledge to work. This is a substance that alters brain operation, and we only have one brain.
Fair enough. I think that I always struggle a little bit with where to place the findings. So what I'm finding are significant differences. I try and use more like mild or modest because the effect sizes are small, and also from like neuropsychological testing, they're generally in either the average, low average, or mildly impaired range. So I think it's important that, in describing the results, that it's measurable, it's statistically significant, and it's in this, it's to me like mostly reduced efficiency and not kind of mildly diminished performance. And I think that, most people that I talk to really take that in and take that to heart. And when I talk to teenagers, including my own son, I really try and say, like, it's about, kind of you reaching your full potential, and this is getting in the way. I also do think that there's some recovery of function with abstinence. We really need better data on that to really make sure it's full recovery, and that, children who started using adolescents get back on their full trajectory. But, that message of hope is useful because it might motivate people to actually quit. Try quitting for a month and see if you notice that you're thinking more clearly, and that your emotional regulation is better, and maybe your stress response is better.
A follow-up, I guess, from Tom. When a child with ADHD is treated with cannabis, in my experience, parents can be the early adapters. Who is really being treated, the child or the parent/educator?
That one's a stumper for me. I think that, I wonder if the sub message there is that maybe if people are treated with cannabis for ADHD, that maybe it's just calming them down, and especially treating that kind of hyperactivity and irritability, and maybe they're received better by parents. I don't know if that's kind of the point there. But I do think we need better data before people are recommending, especially cannabis that's high in THC as a treatment for ADHD. I don't do medicinal research, so I really have to kind of give that caveat that I can't really speak to prescribing and clinical outcomes in that way. But when we're looking at the neurocognitive effects in the research side with recreational use, it does look like it's not helping with their ADHD symptomology or things like complex attention and executive functioning. So it's good for parents to really understand that, because this stuff is really complex, and the information that's out there on the internet and just talking with friends and things like that, they're going to get a lot of messaging that's really goes against this.
I think that's all of the questions in the Q&A section. There was one from the chat from Rahima about sharing the slides. We are recording, and we're hopeful that if technology works on our favor, we will be able to post onto our YouTube channel, hopefully next week, this presentation. I'm not sure about the sharing of the slides separately from that. Dr. Lisdahl.
Yeah, sure, I can
Okay.
do that. I can share that.
So we could also then post a link to those on our website as well.
Yeah, I see another question popped
Yep.
up from Sue. Is there a percent of THC that you'd recommend to be below for a teenager who uses medical cannabis? You know, I feel like it's a little outside of my expertise to give that exact cutoff. What I'll say is that there's a dose-dependent relationship, and there's lots of evidence on the animal and human side that lower potency products or products that have lower THC and higher cannabidiol might be safer to use for neurocognitive outcomes. So, you know, I would love to see more clinical trials that really test that for specific disorders across specific age ranges, but that's my best advice there.
Well, thank you so much for a wonderful talk today. That was so instructive, and I think really highlights a lot of the nuances when it comes to cannabis and cognition. It's not as simple as you know, harm for everyone or absence of harm. It's a matter of where and by what dose, and you know, the interplay with other intervening moderators. So it's a complicated topic, but a critically important one.
Yeah, and I didn't get to go into this, but other research has shown that, you know, in medical users with really complex neurologic and medical diseases, there actually can be some improvement in cognitive function with low dose THC product. So it really does depend on what we're talking about here. You know, the details matter of that potency and who's using and how old are they. And if it is treating, you know, chronic pain or neuropathic pain, you might actually have a boost in cognition because those things negatively affect cognition too.
Great point.
Yeah, thank you so much for having me. I really enjoyed sharing the information with you.